Hi, my name is Colleen McClung. I'm a professor of psychiatry at the University of Pittsburgh School of Medicine. And I'm really excited to share some of our work today and really thrilled to have been a recipient of the Colvin Prize for Outstanding Achievement in Mood Disorders Research. Really a thrill to get this, and especially from the Brain and Behavior Research Foundation, which has been such a great supporter of our work and the work of so many other scientists over the years. So today I'm going to be talking about circadian rhythms and how they're important in bipolar disorder. So I'm going to start with a little bit of a background on circadian rhythms. So circadian rhythms are prevalent in every process in the brain and the body. So basically everything that you do has a 24-hour rhythm. These include things like sleep-wake cycle, of course, but also hormone release, blood pressure, cognition. All of these things revolve around a 24-hour cycle. And the master pacemaker in the brain that controls these circadian rhythms is located in a part of the brain called the suprachiasmatic nucleus, or the SCN. This is kind of at the back of the brain and the base of the brain. And it's responsive to direct light inputs from the retina. And that's what really sets those rhythms in the brain and throughout the rest of the body. So the light comes in through the retina. It will tell the SCN what time it is. The SCN will then regulate all these clocks and regulate rhythms throughout the entire body. So we know that every cell in the body basically has its own clock, and without the SCN, these would all go running off in their own cycles. But because we have the SCN, these clocks are all synchronized to each other over the 24-hour cycle. And they're basically, these clocks consist of these proteins that control expression of genes in the cell that need to function at different times of day. So genes that are involved in energy metabolism or other cellular processes. This circadian clock that functions in these cells really is what sets the time of all of these processes. And of course, if this goes wrong, then you can have disruptions to your system. So environmental disruptions that change the circadian clock can lead to things like jet lag or sleep problems. And endogenous disruptions to this system can also contribute to things like mood disorders or risk for substance abuse and other psychiatric diseases. So we're really interested in understanding, how this clock is involved in regulating all these processes in the brain and in the body, and what happens when this clock goes wrong, and how this contributes to particularly in our case, the development of psychiatric disorders. So one of the diseases we've really focused on a lot is bipolar disorder. And bipolar disorder is a disorder where people have a major disruption to their sleep, to their mood cycles. So they can have phases where they feel manic, where they have increased energy, elevated mood, lots of thoughts, racing thoughts, decreased need for sleep. They can also have times when they're depressed, and so they feel very disinterested in things that normally would give them pleasure. Also just feel very, very fatigued, very low. And actually, these cycles of bipolar disorder are actually kind of similar to cycles in the sleep-wake system, where when people are manic, they have this kind of increased energy where they just feel awake and don't need to sleep. But when people are depressed, they feel very down and like it takes all their energy to just get out of bed. And so there's been some suspicion for a long time that the circadian system was really a key underlying feature or factor that really controls these moods and these changes in mood state. But nobody really understood the mechanisms that underlie this. And here are just some of these examples of why we think circadian rhythms are involved in bipolar disorder. So we know that shifted and blunted rhythms and a delayed chronotype are strongly associated with bipolar disorder. We know that things like shift work, jet lag, seasonal changes can trigger episodes or exacerbate episodes in people with bipolar disorder. So someone might be going along fine and then they work a different shift, or there's a change in season, and this might contribute to the onset of an episode. We also know that polymorphisms in circadian genes, so variations in the genetics of the system that controls circadian rhythms, is associated with bipolar disorder. This has been seen in a number of genetic studies, even genome-wide association studies. And as I'll talk about at the end, we know that targeting the circadian system can be therapeutic for the treatment of bipolar disorder. And this includes a lot of different therapies. And that, like I mentioned, I'll go back to this at the end. So we really got into this field because of a mouse. And so this was a mouse that had a mutation in one of those core circadian genes, a gene called clock. And this was a mouse that was originally found in a genetic screen by Joe Takahashi, Fred Turek, and Martha Vita-Turna. And this mouse has a mutation in, that causes this gene to basically be non-functional. And so if you look at the circadian rhythms of this mouse, this over here on the right-hand side is wheel running activity. Normally mice are active during the night and they're sleeping during the day. And you can see that they have these very regular sleep-wake cycles if you look at their activity on a running wheel. And if you put them into constant darkness, they maintain very strong activity. But these mice actually had a very long period in their activity, so their rhythms were a little bit longer than they typically would be. And over time, they just kind of lose their rhythmicity altogether. And so we knew that this mouse had disruptions to circadian rhythms, but our lab really took this a step further and characterized, excuse me, these mice in a lot of different behavioral assays. So when we looked at their activity levels, of their sleep, of their mood state, their impulsivity, risk-taking, reward value for substances, we found that these mice actually look very similar to people who are suffering from bipolar disorder, and particularly in the manic state. So the mice are very hyperactive, they sleep less, they tend to be very impulsive, they find everything more rewarding. And when we give these mice drugs like lithium or valproic acid, which are first-line treatments for bipolar disorder, we find that this reverses a lot of their manic-like phenotypes. And so this mouse really was our starting point and kind of tying us into looking at how circadian rhythms and how circadian genes might be controlling mood and reward in the context of bipolar disorder. So because these mice had these phenotypes, we decided we wanted to look at brain reward regions. So these are consist of the ventral tegmental area, which is where a lot of the dopamine neurons are housed. Dopamine is a molecule that's really involved in mood and reward and salience for things. And these neurons project to other regions of the brain, like the nucleus accumbens, prefrontal cortex. So we really wanted to focus a lot on this system because of the phenotypes led us to suspect that these circuits might be disrupted in these mice. And indeed, when we measured dopaminergic activity, so just kind of how much those neurons fire, those that contain dopamine in those mood and reward centers of the brain, we found that they have an increase in dopaminergic activity, and this could be rescued with chronic lithium treatment. And so this led us to suspect that maybe a lot of their behavioral phenotypes that look like mania are associated with this increase in dopamine, increase in dopaminergic firing. So clock is expressed all through the brain, all through the body. We didn't know at the time if clock was even in these cells, in these dopamine cells in the VTA, or if this is where it's having an important action. And so we took an approach where we just manipulated the expression of clock in those dopamine cells. And we found that indeed this led to this increase in dopamine cell firing. So this was really one of the first times that we found that the clock gene was really functioning outside of that central pacemaker of the SCN and was actually involved directly in controlling these cells that fire and release dopamine. We went on to collaborate with Kafui Dizaraza at Duke to do some in vivo recordings, so recordings in the mouse. We found that, interestingly, this dopamine cell firing was not really the same all the time in the clock mutant mice. It seemed like it was really happening, this increase in firing was really happening more during the daytime, which is, as I told you, mice are nocturnal. They like to sleep during the day. And so the fact that they had this real increase in dopamine cell firing during the day when they're supposed to be asleep, which kind of then went sort of back to normal during the night, led us to suspect that what's really going on in these mice is that there's a change in the timing of when dopamine is available. And so when you have dopamine being fired when animals are in the wild and they're finding things that are rewarding or finding things that are stimulating, that's okay. But when you have these surges of dopamine that happen at the wrong time of day, when the mouse is supposed to be asleep, this is when you really get problems. And we went on to figure this out in a variety of ways. And so this, again, tying it back to human bipolar disorder might be relevant for why people have these mood episodes when they have a disruption to their circadian system, their normal sleep-wake cycle. They might be getting dopamine at the wrong time of day, these surges of dopamine, and that's leading to this sort of pathological response. So we've gone on from there to really characterize a lot of how clock is working at the molecular level to regulate dopamine. I'm not going to really go through these, but we found that basically clock is interacting with some metabolic factors that are necessary for controlling the energy state of the cell. And so clock is really intimately involved in regulating this very important system that's controlling mood and reward. It is doing so in a way that's reactive to the environment. So we wanted to transition our work into human brain, and the reason why we wanted to do this is because you can only go so far in mice. Mice don't have complex psychiatric disorders, and so we really wanted to understand what was going on in the brain, in the brains of people who actually suffered from these disorders, including bipolar disorder, schizophrenia, depression, etc. And the way that we did this was by taking postmortem brains. So these are brains that are donated by family members after someone has died. And if we know the time of death and the date of death, we're actually able to put them on a 24-hour scale, basically, and do gene expression studies across those 24 hours. And this was actually a technique that was originally developed by Huda Keel and Biff Bunny's lab, and we've been able to use a similar technique to look at brain regions like the prefrontal cortex, striatal regions, and look at rhythms in gene expression in the brain. And what we first looked at core circadian genes, and we found to our surprise and delight that actually we got very consistent rhythms in gene expression, whether our genes were in Pittsburgh, our brains were in Pittsburgh, or their brains that were collected in California. We were able to, when we compared gene expression rhythms across different data sets and cohorts, we saw very similar results. So we can reliably measure circadian rhythms or diurnal rhythms in the human brain across different brain regions with pretty remarkable consistency. And so I'm going to tell you about some results that we had from the striatum. And the striatum is a region of the brain that receives dopaminergic input from the VTA and from and basically the different regions of the striatum are involved in very complex and meaningful actions, including emotional regulation, cognitive control, reward, movement, things like this. And so we separated out the striatum from the human postmortem brain. And we looked at regions, the nucleus accumbens, the caudate, the putamen, which are all regions of the striatum. And again, we found that they had very nice rhythms and gene expression when we looked at core circadian genes. So we wanted to look at subjects that had psychiatric disease, and we decided to focus on psychosis, which is something that can be found in both bipolar disorder and schizophrenia, as well as major depression, Alzheimer's, some other diseases as well. The reason we wanted to focus on psychosis is because the striatum has been strongly implicated in the development of psychosis. And so we were able to get a cohort of subjects, 34 subjects with psychosis. Some had bipolar disorder, some had schizophrenia. We also had non-affected comparison subjects. We were able to match them based on clinical characteristics, including gender, race, age, etc., and do RNA sequencing, so looking at large-scale gene expression, and measure rhythms in these brains. And what we found immediately was that people with psychosis generally had a loss of molecular rhythms in some of these core clock genes. And you can kind of see this here with a couple of examples of Cry1 and Reverb alpha, where in all three regions, the nucleus accumbens, the caudate, and the putamen, subjects with psychosis really just have far less organized rhythmicity in these genes. We also wanted to compare rhythmicity across regions, and it was kind of interesting because we expected we would see a lot of consistency in genes that were rhythmic in control subjects across, for example, the nucleus accumbens and caudate, or the nucleus accumbens and putamen. And this is, here we're doing an approach which is called RRHO, which just allows us to kind of compare two data sets to each other to see how common they are. And we found some similarities, but also a lot of differences. But in the subjects with psychosis, one of the really interesting things we found was that the caudate and putamen, these kind of dorsal striatal regions, tended to sync up. And so what this really suggests is that there's something about psychosis which is kind of synchronizing rhythms across these two regions. And when we look at what these genes might be that are synchronized in rhythms, they tend to fall into classes that suggest something about receptor signaling. And it's kind of tempting to speculate that maybe this has to do with dopamine receptors, which are cyclic AMP activating or inhibiting receptors, G protein coupled receptors. So it could be that people with psychosis who are known to sort of, or thought to have this kind of excess dopamine, are, it's creating this kind of synchronized state across the striatum. It's also possible that antipsychotic treatment is somehow leading to this synchronization, but it's something we're really interested in following up on. And we think that these human gene expression studies are really going to be really exciting for us to move forward with in terms of trying to understand the underlying biology of what's going on in the human brain in terms of molecular rhythms. So as I mentioned, I wanted to sort of end with therapy. So our lab is very invested in trying to understand how current treatments for bipolar disorder are working, and maybe look at the circadian clock as a way of identifying new treatments that could could really be beneficial for people with bipolar disorder. And it turns out that actually most treatments for depression and bipolar disorder affect the circadian clock in some way. So of course there's treatments like bright light therapy, total sleep deprivation, which is used as an acute antidepressant, social rhythm therapy, which I'll show you in a minute. This is a therapy, an interpersonal social therapy where behavioral therapy where they put people on very strict sleep-wake and social schedules. And this tends to really synchronize their rhythms, as well as medications that are currently used for antidepression and for mood stabilization. So here's an example of someone with bipolar disorder on the left here. This is actigraphy, so measuring activity using a device like this. And you can see that when you measure in this individual's sleep-wake cycle, they have very irregular sleep-wake patterns over a couple of days when you plot them over 2 days over the course of weeks. And over here is their mood. You can see negative is depressed, positive is manic, and you can see that this individual's mood is also kind of all over the place. But when you put them on a very strict sleep-wake cycle where they're sleeping at the same time of day, getting up at the same time of day, not only does it stabilize the sleep-wake cycle, but it also stabilizes mood, which suggests that if you can really amplify this circadian system, then this might be therapeutic in the treatment of bipolar disorder. So with the clock mutant mice, going back to our mice, we've been looking at how current treatments are affecting the circadian clock and affecting their behaviors. So valproic acid, Depakote is a current treatment for bipolar disorder. We found that it is able to reverse their behavioral phenotypes in a number of different assays. So it seems to be therapeutic in these mice. But it also increases rhythm amplitude, basically. And it turns out that lithium kind of does the same thing. So these are experiments where people put a reporter gene into cells that allows you to measure molecular rhythms over the course of hours or days. And what you can see is that if you give these cells lithium or you give these cells valproic acid, it kind of strengthens those rhythms. It makes them, have increased peaks and decreased troughs. So really increasing that rhythm amplitude. We also see this if we use cells that are taken from the clock mutant mice. So if we give these cells valproic acid, we're able to increase the rhythm amplitude. So what this suggests is that valproic acid is actually able to increase these rhythms, even in a situation where you have a genetic mutation that's affecting the circadian clock. And so, so we think that valproic acid may be really beneficial in this way in terms of stabilizing rhythms. And in fact, to take this one step further, if you give clock mutant mice valproic acid and look at their activity rhythms, you can actually see that not only does it synchronize their cellular rhythms, but when you look at wheel running activity compared to clock mutants that either have a long period in their activity or kind of a rhythmic activity, you can see it really stabilizes their activity. So in the short term, what valproic acid might be doing is decreasing dopamine. So basically just acting as an anti-manic agent in the short term by decreasing dopamine. But then over the long term, as a mood stabilizing agent, it may be doing this through increasing these rhythm amplitudes similar to social rhythm therapy and really, kind of synchronizing that circadian clock and keeping it in lockstep and keeping those mood episodes in check. So we've gone on to collaborate with some pharmaceutical companies. We've used drugs. This is a casein kinase 1 inhibitor. So this is a drug that impacts the circadian clock directly. And we found that in the clock mutant mice, it could normalize their behavior in a number of behavioral assays, including things like the elevated plus maze and forced swim test, dark light, which are measures of kind of exploratory drive and mood-related behavior. And this drug's actually gone into clinical trials for treatment of bipolar disorder. And so we think drugs like this that companies are really interested in could be coming along and going to market, or at least going into clinical trials in the very near future. And we're also collaborating with Jake Chen in Texas, as well as others who are developing new compounds that affect the circadian clock in a variety of ways. And we're very excited about this possibility of creating novel medications that could stabilize rhythms in very particular ways. And so therefore, maybe you'd have some of the really beneficial therapeutic effects of drugs like lithium and valproic acid without having a lot of the side effects that come along with that. And they can be tailored to a person's individual rhythms. So if you know someone's rhythms going in, you might be able to give the appropriate medication to kind of get their rhythms back to where they need to be. So in conclusion, I've showed you that circadian rhythm disruptions are strongly associated with bipolar disorder and represent a key feature of this disease. Mice with a mutation in the clock gene have a phenotype that resembles bipolar disorder. A lot of what's causing this seems to have to do with their dysregulation of dopamine, not only just increased dopamine, but having dopamine at the wrong time of day. And so this seems to be really key to the development of these phenotypes that we see. And these mice have really provided us with a great insight into how circadian genes control mood and those mechanisms that really link that circadian clock to mood disorders, particularly bipolar disorder. I've showed you that molecular rhythms can be measured in human brain. And this has given us a lot of insight into the diseases themselves and how rhythms are disrupted in the brains of subjects with these disorders. And we think that long-term mood stabilization can occur via stabilization of rhythms, and we're really excited about the development of novel compounds that could help become the next therapeutics. So with that, I will thank the members of my lab who contributed to this work and our collaborators who help us a lot, particularly with the biostatistics and the human brain studies. Of course, the families who donated brains to these studies, they can't be done without those families. So we thank them, and our funding, and in particular, the Brain and Behavior Research Foundation, which has been very supportive over the years. So thank you very much.